This video is about DNA, or deoxyribonucleic acid, which is present in nearly all cells and contains the genetic information of an organism. It's the information required to produce new cells and to tell the cell how to work. DNA is a polymer made up of 4 different nucleotides, and each nucleotide has 3 different components. It contains a phosphate ion, a pentose sugar, which contains a 5-membered ring, and a base. You do not need to know the structures of all of these components of the nucleotide, as they are given on the data sheet. What you do need to know is how they join together to form the nucleotide. The phosphate ion shown on the data sheet is really a dihydrogen phosphate ion, so H2PO4 1-. The sugar is a deoxyribose sugar, and that is because the highlighted carbon at the 2 position doesn't have an OH group attached to it. Stop the video and use the data sheet to add the names of each of the 4 bases that are included in the nucleotides. The 4 bases are all examples of heterocyclic molecules. That's ring structures containing a mixture of carbon and other atoms. In all 4 cases they contain nitrogen atoms as well as carbons in the ring. Nucleotides are formed by condensation reactions. That's when 2 molecules join together and a small molecule is eliminated. The phosphate ion binds to the sugar and water is eliminated, and the organic base binds to the sugar and water is eliminated. The diagram shows a simplified structure of the nucleotide. The phosphate is bonded to the sugar, which in turn is bonded to the base. The diagram shows the structure of the adenine nucleotide. The phosphate group is linked to the sugar via the carbon at position 5 on the diagram, and the base is linked to the sugar via the carbon at position 1. Make sure that you don't draw a straight line directly from carbon 4 to the oxygen of the phosphate group. There's another carbon, carbon 5, between carbon 4 and the oxygen. There are several nitrogens in the rings of the 4 bases. In each case, it's the nitrogen which is in the NH group at the bottom of the molecules shown on the data sheet. This is the nitrogen that becomes bonded to the sugar at position 1. The bottom diagram shows the thymine nucleotide. Again, look carefully at where the base is joined to the sugar. That's at carbon 1, and where the sugar is bonded to the phosphate group at carbon 5. Make sure there's an additional carbon between carbon 4 and the oxygen of the phosphate group. In the exam, you may be asked to draw the nucleotide in a different orientation, a different way round. You will be allowed a piece of tracing paper in the exam to draw the diagram from the data sheet and then rotate it so that it's in the correct orientation for your diagram on answering the question.